Well, good afternoon. I'm Susan Sheehan, President and Chief Operating Officer of Huntsman Cancer Foundation, the philanthropic entity that supports all the wonderful work that's happening at Huntsman Cancer Institute. And it is really my distinct pleasure today to introduce to you Dr. Alana Welm. She has a spectacular bio, and I just wanted to highlight a couple of items on that. And that is that she completed her doctorate at Baylor in molecular and cell biology. And then she did her postdoc at a University of California, San Francisco lab in breast cancer metastasis. She came to Huntsman in 2007. And Dr. Welm, I remember the moment in the buzz and the excitement when we found out that you had accepted the offer and were coming. We talked about a very talented couple that were coming to Utah, and that we had poached this superb talent from UCSF. And it was a major victory for Utah, for Huntsman Cancer Institute, and for the University of Utah, because she was going to really be able to take our breast cancer programs to a whole new level. I have been working closely with her since 2007, watching her career and then watching her really make the contribution that we had all predicted in 2007 she would contribute with her husband, Brian Welm, together to understandings about breast cancer. What she's here to talk about today is something very specific, and that's why breast cancer spread and what we've learned about what might be driving that. And then even better, how to really do precision medicine around breast cancer to help better predict who is going to have a breast cancer that metastasizes or comes back, and who will, have a complete recovery with no recurrence later on. And her work has been very focused on that. In addition to being a great scientist, she's an incredible human being who I have watched over the years really spend time in a very patient and compassionate way with people who are personally affected by breast cancer, and explain and meet people where they are in their own questions and fears and need for hope in the breast cancer realm. And she is somebody that I call upon when I have somebody who is just in despair and needs a reason to hold on to something positive. She is that positive. She's a mother of two very talented children. I follow them on Facebook, and I consider her a good friend as well. So we are just so honored to have your time today, Dr. Welm, and I will now leave this chat up to you.
Thank you very much, Susan, for that really, really nice introduction. And I can attest that Brian and I were just as happy as you when we decided to come here to Utah in 2007. And indeed, it's been a fabulous place to carry out our work. And we could have never imagined the types of things that we could do here that we have been able to so far. But I will say that we still have a lot of work to do. So that's what I'm going to talk to you about today, is the next phase of precision medicine in cancer, and specifically in breast cancer, since that's what we work on. And really, this is something I'm really passionate about. So it's one of my favorite topics, but I promise I'll finish within the hour. So just as a start, where are we with breast cancer? So some people need to be reminded about how important this disease is, because many people get breast cancer and are cured from it, which is a wonderful thing. But the fact is that so many people get breast cancer, more than 300,000 women every year. And also, don't forget about a fraction of those cases actually do happen in men. So male breast cancer is real, and men die from this disease as well. So this is a very, very common cancer. And as such, even though we do a pretty good job with curing most people, it actually still causes death in more than 42,000 women every year, and also 460 men every year. And when we think about those numbers, it's hard to put them in perspective, but I've calculated this, and that's actually one person dying from breast cancer in the US every 12 minutes. So every time I give one of these seminars, I remember that 5 people have died in that time of breast cancer. And the problem is that we still don't understand very much about the deadly form of breast cancer. And that's what I'm going to talk about today. And in fact, we haven't really had a true acceleration in the rate of decreasing deaths from breast cancer or mortality since the 1990s. So when tamoxifen, one of the hormone therapies, came on, and Herceptin, a HER2 targeted therapy, this really did change the paradigm. But since then, even though we have had progress and we have had new drugs approved, that rate of decrease is still about the same. And that's actually illustrated in a couple of slides that I'll show you. I wanted to also give you a global picture of breast cancer. So breast cancer is the most common cancer in the world. 2 million new cases in women every year. And 500,000 deaths every year of this disease. So if we keep up our current rate of progress, which I'm going to talk about in the next slide, it's estimated that by 2040, we'll have almost 1 million women dying every year of this disease. So it's really something that I wouldn't say we really have a handle on, even though we have made progress. So this slide is something that is just astonishing to me. These are data from looking at global incidence of breast cancer from the International Agency for Research on Cancer, which is part of the World Health Organization. And I just want to draw your attention to the graph over here on the right. So this is looking at breast cancer across different regions of the world. And in blue is shown how many people per population, so per 100,000 people in that country, are diagnosed with breast cancer. So that's the incidence of breast cancer. And the first thing you see is that the blue bars are larger on the top. And that's really because of increased screening. So in the more developed regions that are shown up here toward the top of the graph, we detect more breast cancer. That's because we do mammography screening regularly. And if you look for more breast cancer, you'll find more breast cancer. But what's more shocking about this graph to me are the red bars. So the red bars are the number of deaths every year for breast cancer. That's normalized to the number of people in each country. So it's the both age matched and population matched mortality rates. And what's really striking to me is that those red bars are pretty equivalent across the whole world. That means that mortality due to breast cancer is not any less in the most developed countries with the state-of-the-art treatments compared to the less developed regions of the world. And the reason for that, I believe, is because we still don't understand the deadly form of the disease. It also tells me that the answer is not all in screening and early detection. I think there was a point when we thought if we just had everyone get their mammograms on time, we would be able to prevent deaths from breast cancer. But the fact is, we find those tumors early, and that's great, but those are the ones that we can cure. The problem are the deadly ones that represent in the red bars. And the deadly form of breast cancer is metastatic breast cancer. And I would argue that this comprises a whole different biology. It's really a different kind of tumor that arises in the breast. It is breast cancer, though. And it's something that we still have to understand if we're going to make a difference in mortality from this disease. So here's the graph I was telling you about when I was talking about how we don't really have an acceleration in progress of reducing breast cancer mortality. This does not mean we haven't made progress. We have made progress and people are living longer with metastatic breast cancer. But if you just look over time since the 1990s, and this graph is separated by race, what you can see is the slope of the line of the decreased mortality of breast cancer is about the same. You know, it's even throughout all these last decades. And that says that we are making progress, but we haven't gotten a paradigm shift. We haven't made a fundamental change that would drop this line down with a steeper slope down to zero mortality, which is really where we want to go. So why is this? Especially given that we are doing more mammograms than ever, we have more early diagnosis than ever. And yet, why do we not have a huge acceleration in reducing death from breast cancer? And as I mentioned, I would argue that's because the biology of metastatic breast cancer, the deadly form of the disease, is actually different, and we don't understand it very well. So unless we get that last piece of the puzzle and understand metastatic disease, we won't be able to have a real paradigm shift here in breast cancer mortality. So what is metastasis? So the first thing you should know is that the vast majority of deaths from breast cancer are because of metastasis. And metastasis is defined as the cancer spreading from the original site of origin, in this case breast, to other organs in the body. And breast loves to metastasize all over the body. Breast cancer does. So it goes to bones, brain, lung, and liver. And when it gets into these organs, that is incurable disease and is the deadly form of breast cancer. So when we look at the statistics, it's about 20 to 30% of all breast cancers that actually are this metastatic type. And even though we don't know they're metastatic, when the tumor gets found and originally diagnosed, those tumors have actually already spread through the body in an undetectable state. And so that the horse is out of the barn, so to speak. And this is the real struggle, the real problem with this disease, because early detection doesn't seem to help. Those tumors can start to metastasize even before they're even detectable. And the other thing to remember is breast cancer is not one disease. We call it breast cancer, but in fact, there are many, many different types of breast cancer, and all of them are so different that essentially every tumor has its own personality. These metastatic cells, once they spread from the breast into other parts of the body, this happens mainly through the bloodstream, but can also happen through the lymphatic systems. They will sit there in these distant organs quietly and undetected. And they can hide out like this for years or even decades before they actually start to grow again into another tumor, which would be then diagnosed as recurrent or metastatic or stage 4 breast cancer. And this period of time where they're undetected and hiding out in these organs is called dormancy. This is a period at which there's no evidence of disease, but these cancer cells are in fact still there. The problem is we can't see these cancer cells. We can't detect them even using the most state-of-the-art imaging or blood tests or anything like that. And therefore, we know almost nothing about these tumor cells. Why do some of them grow a few years later? Why do some of them not grow? How do you study something that you cannot even detect or see? Unfortunately, when they do grow into a metastatic or stage 4 tumor, the disease now is incurable. And it is very treatable. At that stage, we have many therapies, I'll show you in a minute, that are approved even for metastatic breast cancer that do extend the life of the patient, the time, but they don't cure the patient. And the patient will almost always inevitably die of their disease eventually. And why is that? Why is it that when it's in the breast, we can cure it? Why is it that when it spreads to other places, we're not able to? So we really need research that's focused on how to treat this metastatic disease and also how to personalize therapy. So to date, one of the problems is that the main focus in research has been on primary tumors. The reason for that is we have, as I said, something like 240,000 breast cancers diagnosed every year in the US. Those patients always undergo surgery. It's easy to get those tumors. They're doing surgery, taking them out and throwing them away most of the time. And so it's very easy to get those tumors and do research on them. But now with the realization that we have a growing need to study metastasis, those efforts have increased. However, it's still challenging. The reason why it's challenging is, we can't do research directly on human patients most of the time. That's obviously limited for ethical reasons, except in the context of very structured clinical trials. So getting these patient metastasis samples are really difficult. Not many people want to just have a biopsy of their lung or their liver to give us a piece of their metastatic tumor. And so it makes it much more difficult to study. It does require sophisticated model systems because the very definition of a metastatic tumor is that it can spread through the body to different organs. We really have to try to study this in a whole animal system if we can't study in the patient. And so we typically use mouse models for that, which are quite time consuming and expensive to study. We are getting new therapies approved for certain types of breast cancer. Some are lagging behind, and this is in the metastatic setting. But the benefit of these therapies is limited, meaning, again, the disease still is not curable, although we are having people living longer and longer with these new therapies. Toxicities are a problem, whether we're talking about hormone therapy, chemotherapy, or other agents. Immunotherapy, which I'll come to, they all do have toxicities. And of course, treatment of this disease, the cost is high. So if we want to improve mortality rates for breast cancer, we really have to find better treatments for metastasis and also prevent that from happening in the first place, or prevent them from growing out, even if they've already spread at the time of diagnosis. If we can keep them dormant, then that would be a big win. So what are our other advances? I do want to mention immunotherapy because that has been something that has been, was breakthrough of the year in 2013 and has been really exciting in many other types of cancer. And the way that this works essentially is through the discovery that the patient's own immune system can be activated in order to hunt out those cancer cells and kill them. So even if we can't see those cells, our immune system can see those cells. And maybe we could figure out a way to get those immune cells to kill the hiding cancer cells. And actually, immunotherapy approaches have made astonishing advances in certain cancers, most notably melanoma, which at that time, stage 4 melanoma was invariably deadly in a very short amount of time, as well as small cell lung cancer and other lung cancers, where now these diseases are actually curable with, in some cases, with immunotherapy. Unfortunately, in breast cancer, immunotherapies are not working very well. We do have one particular therapy that's approved in one type of breast cancer that's called atezolizumab. It's approved in patients who have so-called triple negative breast cancer. That means it's negative for hormone receptors and negative for HER2. And that's approved in combination with a certain type of chemotherapy. But unfortunately, we haven't seen a very big improvement in overall survival because very few people actually have a good response to immunotherapies. But because of the excitement, there are still thousands of clinical trials trying to figure out how to make immunotherapy work in breast cancer. And most of those involve combining this drug with various other types of traditional therapies. So we'll see how that goes, but I don't have very high hopes yet that is going to be a breakthrough. We really do need more research. The important thing about immunotherapy, though, is if we can figure it out, we could apply it to both prevention and or therapy. So I'm actually involved with a national effort called the National Breast Cancer Coalition Artemis Project, which is focused on both preventing breast cancer in the first place, as well as preventing metastatic disease. And we're really looking at immunotherapies in both of those settings. And actually, we hope this year in 2021, we're going to have a trial of a new vaccine to prevent breast cancer in the first place. So I'm really looking forward to those studies. So what are the research questions? So as I mentioned, most breast cancer research is done on primary tumors, so those that are from the breast, or in mouse mammary tumors. And that's so that we can do research outside of humans, but still have a whole animal model in which to study the metastasis process. And some research is also done on human breast cancer cell lines that have been cultured for many decades under non-physiologic conditions. So in essentially a petri dish in an incubator in the lab. Maybe some of you have heard of the book, The Immortal Life of Henrietta Lacks, that talks about the development of the HeLa cancer cell line. That was not a breast cancer cell line. It was a cervical cancer cell line, but it's the same kind of concept. So the biology of metastatic breast cancer, as I said, is still a mystery. And it's a mystery with regard to those tumor cells that spread and why they're different and what makes the ones that can wake up and what makes some stay dormant, but also the tissue environments that foster them. So it actually turns out there's a really nice sort of interaction that has to happen between, say, the normal liver and the cancer cell that gets to the liver in order to allow those cells to survive and eventually grow as metastatic tumors. So we desperately need access to fresh metastatic breast tumors for research to avoid the pitfalls of some of these breast cancer cell lines, or we're really good at curing tumors in mice, but that's not our end game. And so we need biopsies from patients. We also get fluids from patients who have a metastatic tumor that causes their lung cavity to fill up with fluid and it needs to be drained. We can get cancer cells from that. And we've also started getting autopsy material for patients who want to donate their bodies at the time of death for research. And that is a sensitive issue, but really is a game changer in being able to understand the lethal form of the disease. Also, new therapies really should be discovered and validated in the metastatic setting. That's what we're trying to cure, is metastatic breast cancer. So we should test them against metastatic breast cancer. And the best models for this, we're convinced, are those that come directly from patients. And that's what I'm going to talk to you about, because that's kind of the thing that my lab started when we first came to Utah. And we hope that anything we find there in terms of discoveries or new drugs would have direct relevance to the clinical trial in which that drug or approach would be tested. So here's just a little schematic of how we go back and forth between the laboratory models and the lab. So these would be cells in culture in those petri dishes, our mouse models. and then of course into the clinic with real patients. So here you see an example of a breast tumor shown on mammography. And then in this case, it has spread to the bones, which lights up the spine. Breast cancer really loves to live in the spine, or these tumors that are in the liver. So the key points here are that we really do need models that are most like the patients. So slow growing, spontaneously metastatic human breast tumors in order to test new therapies. We want to study this on all the main types of breast cancer. As I mentioned, there's many types of breast cancer, but we don't want to leave any one of them out. And one of the things that we struggled with when we started our lab is that these breast cancer cell lines that people have made and worked with for so many years, you can study metastasis with them if you then, you can inject them into the mouse breast, the mammary gland of the mice. They'll form tumors and they'll metastasize. But the problem is that these human cancer cell lines are actually pretty bad at metastasizing in the mouse. And it seems like they've maybe forgotten how to interact with their environment in a way that facilitates sort of this liver tumor interaction or bone tumor interaction, as I talked about before. And so that really prompted us when we started our lab in Utah, and actually with great support from the Huntsman Cancer Foundation for our initial funding, to be able to take these tumors out of patients and just put them directly into mice. And this is called a xenograft. So we're grafting tumors from one species into another in order to make a living model of that person's breast cancer. And that was a completely new approach for breast cancer, and in the particular way we did it. And so that has been, I think, something that's really made the mark for some of the things we're doing here at Utah. So our first study came out in 2011 in Nature Medicine, and here we reported the first sort of big bank of these, what we called at the time, tumor grafts that were derived from women with breast cancer. We did both primary tumors as well as metastatic disease. And we grew these in special mice that don't have functional immune systems. And then we were able to study that disease. And what we reported in this paper was that these tumors actually looked very much like the original human tumor, and they even metastasized in the mouse, very similar to what happened in the tumor patient. Since then, we've continued to work with these so-called PDX models, patient-derived xenografts, but we've also extended this work largely in collaboration with Brian Welm's lab, my husband, in making, skipping the mouse work and actually doing this in three-dimensional gels in the lab, which kind of mimic the environment of the organ without actually needing a live animal. And here these, what we call organoids, grow in these clusters inside the gel. And so we've been able to utilize those for quite a bit of the work as well. So why, when we realized we could grow patient tumors in this way, and we got a big bank of them, we started wondering, well, maybe we can use these to have greater impact. And this is just a quick summary just to remind people of what a typical breast cancer patient would go through. And that is, originally getting diagnosed after finding a lump or a lesion on a mammogram. And then they have to undergo surgery to remove the primary tumor in the breast most of the time. And then a systemic therapy as well. And this can include either hormone therapy, chemotherapy, it could be HER2 targeted therapy if it's a HER2 positive tumor. And that therapy can happen either before or after surgery. It sort of just depends on the scenario. And then after surgery, if more chemotherapy is given, that's called adjuvant therapy. And the whole point of giving that therapy after the tumor is completely removed from the breast and there's no evidence of disease is to prevent those disseminated cancer cells that have spread from growing into metastatic tumors. So it's sort of a preventative strategy for metastasis. But unfortunately, despite all of these therapies that we have approved for that prevention, we still end up with 20 to 30% that will end up having their tumors come back in a distant organ like the liver, lung, bone, et cetera. The type of therapy is largely driven by the type of tumor that one gets diagnosed with. And the most common type would be these hormone receptor positive tumors called ER positive, PR positive tumors. They express the estrogen and progesterone receptors, and that's great because this means that these tumors are typically responsive to hormone therapy, where if you block estrogen, those tumor cells have a hard time living. Another type would be the HER2 positive breast cancers. These express a special protein on the surface called HER2, and HER2 is a protein that can be targeted with particular therapies like Herceptin, which is also called trastuzumab. And now we have different variations of HER2-targeted therapies that are effective in these patients. But once the tumor comes back, so the patient at this time has already gotten their therapies, their hormone therapy, their HER2 therapy if they have HER2 positive cancer. Many times they've already had chemotherapy, and yet these cancer cells came back. So then what do you do for treatment? Well, it turns out we have something like, I think it was 37 or 38 therapies at last count that are approved for metastatic breast cancer of different types. And so that's on one hand great, because we have lots of therapies that can be chosen to treat these metastatic breast cancer, and some of them will work for quite some time. The challenge, though, is that we have many therapies, and the selection of which therapy to use, while still largely driven by the type of the tumor, often, especially when it comes to chemotherapy, is often actually kind of a trial and error approach. So if you're doing trial and error with relatively toxic drugs, to me, we might be losing time. And so what we thought about is, well, if we can grow people's tumors, then maybe we can personalize their therapy. Maybe we can actually figure out which of these therapies works on that particular person's tumor, and pick the ones that work. And that we call this functional precision medicine, because we're actually functionally testing each of these drugs on the patient's tumor to see which one works. And the obvious idea would be that we could pick a therapy that is more effective for the patient. But the other important implication is that if we don't give therapies that don't work on those tumors, we would avoid all those toxicities of the trial and error approach when you give a drug that doesn't work, but it still has toxicities. So this is just an overview of our program, where we make these models out of patient tumors. And this could be primary tumors, it could be metastatic tumors. We grow them either in the mice or in the organoids in the gels, and then we analyze them. So the first question we had was, well, how well do these models represent the cancer that it came from? And we do that mostly by looking at mutations in the DNA of the tumors in the patient compared to the ones after they grow in the models, and things like that. After a few years of doing that, we're really convinced that we're growing good models that have high fidelity to the original patient tumor. So then we moved to functionally assessing them. And this really largely has been, can we give drugs to these models and see if it works to shrink the tumors or kill the tumor cells? Oops, sorry. And with that, then we can start at this point to return those results back to the clinic in the context of clinical trials to see if we could actually improve clinical outcomes for those patients. So this is a scenario that we term functional precision medicine. And the reason why I put the emphasis on functional is because we're actually testing the drugs, as opposed to the traditional precision medicine approach would be just sequencing the DNA to see if we can find a therapy that would work on a particular mutation or a protein that comes from a mutated gene. So with this, we've been able to really sort of stand apart in the field where we developed this program, and now we're one of 6 NCI designated, National Cancer Institute designated PDX development and trial centers across the nation. And our goal in this is to have models from 100 patients here at HCI, and also in collaboration with our collaborator Mike Lewis in Houston. And so we get these models from 100 patients. We do the mutation analysis, but we also test the drugs in the models to see if we can find new drugs that might work in breast cancer of different types, different subtypes of breast cancer, but also understand why some cancers respond to some therapies and not others. And the kind of data we get looks like this. This is just a heat map showing that these are different patient tumors across the bottom. And we've given them all a single drug that's still in the experimental setting at different concentrations. So the highest concentration at the top, and then lower concentration. And where it turns red is where we've killed those tumor cells. So we can see, like, okay, about half of our patient tumors were really nicely affected by this drug. Why? And we can use our models to try to understand why. And then now we're just about to take this drug actually into a new clinical trial in triple negative breast cancer, where we see the most activity. So I'm going to switch gears a little bit just to give you an example of how we envision this working in the future at Huntsman. We think that these approaches of making better models and finding new drugs will definitely help in the future. That's our goal. But it really pushes us to say, but what can we do right now? There are people right now with metastatic breast cancer that can't afford to wait for 10 or 20 years for a new drug to come online. And this actually question I'm really passionate about because it's something that I really feel the urgency for. And we actually got a hint that we might be able to do something based on this data. So these data came from the paper I mentioned that we published in Nature Medicine in 2011, where we were just trying to make models from patient tumors. And we were just putting them in mice, seeing if they would grow. And what we found is only about 30% of those tumors actually grow in the mice. That's a little bit disappointing because we want to make more models. But on the other hand, what we found completely accidentally, when we looked back at what happened with those patients, we found that nearly every single patient whose tumor actually grew in the mouse had died of their disease by 5 years. In contrast, almost all of the patients whose tumors we couldn't grow in the mice were still alive. So that hinted to us that maybe by asking these tumors to grow in this foreign environment of the mouse, we could actually predict how aggressive they were. So this was an initial idea, as I said, not expected and very surprising. And so we opened a new trial called Towards Functional Precision Medicine for Breast Cancer. This trial has just completed accrual of 80 patients. And the goal here was to say, could we actually prospectively predict if someone's tumor was going to be one of the bad actors, let's say, that would actually cause aggressive disease versus those that would not, based on whether or not their tumor grew in the mouse. And so we enrolled people who had newly diagnosed breast cancer before they got the therapy. We took a biopsy of the tumor, and then we tried to grow it in the mice. And this is a little bit out of date because, as I said, we just finished our target accrual of 80 patients. But even with this smaller number earlier on, we had really phenomenal results, which was that as we had expected from our earlier study, the people whose tumors grew in the mice had a much worse outcome. And in fact, they had a 44% recurrence rate, meaning their tumor came back as metastatic within the short course of this study. They were 20-fold more likely than the people whose tumors didn't grow in the mice to have a recurrence. So this really said, wow, maybe we can actually start to make predictions about who needs more targeted therapy. On top of that, before I go into that, on top of that, we have their tumor growing. So if we actually think that their tumor is going to come back quickly with aggressive disease, maybe we can do our functional precision medicine to find a therapy that would work for them before their cancer ever comes back. So I'm just going to quickly go through this slide. This is probably a little more detailed, but here's the information in the bottom two graphs that I want you to see. This is looking at how these tumors are coming back as the blue line drops. And these are the ones with the PDX, the mouse tumor grew. You see how fast those tumors came back, where 20% of the patient's tumor has recurred within about 400 days. In contrast, the people whose tumors didn't grow in the mouse are not recurring over this time. And the same is true for survival. Unfortunately, these patients are dying very quickly, usually within a year or so of their diagnosis, if their tumor grows in the mice. So it gives us some predictive power. So we started to ask, okay, well, we have these aggressive tumors. Let's do something about this. Let's see what we can do with the models that we have growing. So the patient, in the meantime, while we're trying to grow her tumor, is undergoing regular therapy. So this is just the standard of care chemotherapy that happens, in this case, before surgery. That takes about 6 months. And then in the meantime, we're busy working in the lab to figure out the right therapies. And then once we have that information, we're able to return that information back to the physician in the context of this research study. So I'm just going to give you one example of a patient on this study. This is patient 19, so we have her information all the way through the course of her therapy. She was diagnosed with a triple negative breast cancer, so no hormone receptors, no Herceptin. She didn't qualify for any of those treatments because she doesn't have the ER or HER2. We got a biopsy of her tumor at diagnosis before she started her therapy. We made the PDX model by growing it in the mouse, and the PDX grew. So that really told us that this might be one of those aggressive cancers. We also made the organoids. So the PDXO are just the organoids that we grow in three-dimensional gel cultures in the lab. In the meantime, the patient was undergoing regular chemotherapy and she had surgery at the end of that. And remarkably, she had no disease at the time of surgery. So she's one of those patients that it really looks like there could be a cure. But that was in kind of in contrast to the fact that her mouse tumor grew. So that had us a little bit worried. When we did mutation and genomics analysis on the tumor, this isn't that important, but for anyone who's interested, it was actually a very interesting kind of rare type of tumor that doesn't express the estrogen and progesterone receptors, the hormone receptors, but actually expresses androgen receptor instead. And this is a hormone receptor that's best known for its role in prostate cancer. So some breast tumors do actually respond to testosterone or androgens instead of estrogens. So that was one clue to this tumor might be different. So unfortunately, despite the fact that there was no tumor left in the breast at the time of surgery, and this patient was, completely disease-free as far as anyone could tell, unfortunately, our prediction was correct in that she did have a recurrence because, which our prediction was based on the fact that the tumor grew in the mouse. Her recurrence was in the liver. So she ended up with liver metastases. And we were blinded to this at that time as part of the study. So she went ahead and got her first line of post-metastatic chemotherapy. And in this case, it was capecitabine. And we pulled her models out of the freezer and started screening drugs to see if we could actually find a drug that might work for her. This is just a heat map showing all the drugs we tested, and which ones they are doesn't really matter for the purposes of this. And I'll just say that the dark colors mean that the drug is working better, and the light colors mean that the drug had no effect. So, and the arrows down here just are drugs that we either chose to pursue because we thought they would be good candidates, or they were drugs that she got as part of her standard clinical care. So for example, the chemotherapy she got before surgery is the doxorubicin and the paclitaxel. And you can see that those are pretty potently effective against the tumor, which is consistent with the fact that her breast tumor disappeared on that chemotherapy before surgery. But we actually found a drug up here marked in red that looked really potent against this tumor called eribulin. And eribulin is actually FDA approved for metastatic breast cancer. So we got that, it kind of caught our eye right away. And what was remarkable is when we tested the drug on the mouse tumors, we could see that the eribulin was the only drug that could completely shrink this tumor away. We also tested the prostate cancer drug enzalutamide because of the androgen receptor, and that did slow the growth of the tumor, but it wasn't able to shrink it the way eribulin did. So moving forward back to this timeline with the patient. So the patient, unfortunately, the capecitabine was not effective in this setting. She then went on an immunotherapy trial at that point because everyone wants to try immunotherapy in case it will work magically, like it does for a few people. But unfortunately for her, that was not very effective. We had returned our functional drug test back to the physicians at that point, and they did then, after this immunotherapy didn't work, they did put her on eribulin, and we were really excited to see that she had a complete response to this drug, and all of her liver metastases completely shrank away. All the fluid disappeared. It was really, really exciting for quite some time. This is over a period of months. Unfortunately, though, eventually, as I said, metastatic breast cancer is still not curable, and she ended up with progression in the bone. So the liver was still fine, but now we had a new tumor growing in the bone, which caused the eribulin therapy to be stopped. And instead she got radiation to treat that bone tumor. And then over time, without any systemic therapy, without the eribulin, the liver metastases unfortunately came back. And then the next therapy also was not effective. And unfortunately, this patient did die of breast cancer. So this is not a win. We still have a lot of work to do. But what I would say is that we're very happy happy that the drug that we were able to test and personalize did give her the longest survival time of all the other therapies. And so we hope that if we could do this earlier and better, maybe we could have better outcomes for the patients. So how do we implement this type of idea in the clinic? This is really a first. Nobody is doing this right now in breast cancer. And so we're kind of paving the way, which is both very exciting and also very challenging. So we're developing the infrastructure to do this. We need to do it fast. We have to be faster than the tumor. And we need to see if the doctors actually, for lack of a better word, believe our data. and this is a really unconventional approach to take. And so for that reason, we just opened a couple months ago a brand new clinical trial called FORESEE, which stands for Functional Precision Oncology for metastatic breast cancer. And it's exactly for this reason, to test feasibility of this functional precision oncology approach in metastatic breast cancer. So the way this works, it's a very small trial, only 15 patients. The way it works is that when someone has their breast tumor come back as a metastatic recurrence, we will get a biopsy of that tumor. We'll grow it in the lab as organoids. We also do the DNA sequencing to look for mutations that might be targetable. And then we screen for drugs. And all this happens while the patient is undergoing their first line of standard therapy in the clinic. And then the idea is to get drug screening done on this tumor model within 12 weeks. And the reason for the 12-week time period is that's a typical interval when the PET scans or CT scans happen to see whether the drug is working. And if the patient has progression, it's time to change therapy. And so what we hope to do is at the time when the therapy needs to be changed, we give this information back to the physician, who then can choose whether they want to keep going with standard regimens, standard therapies, or whether they want to switch to something we've told them in the precision therapy realm. I will say that this decision is not mandated by the study. So because it's still research study and the work we do in our lab is not an official, what we call CLIA certified laboratory test, it's just research. The physician is not obligated to use the information we give them. They can make their choice. And so actually part of the interesting part of this trial is really the goal is feasibility. How often can we make a good model from a patient's tumor? How often can we get a drug that looks effective within 12 weeks? And how often does the doctor decide to use that information? So it's really a first, and we'll see what happens. I'm really excited to see where it goes. And this is being led by Dr. Chris Vaklavas here at HCI. So just to summarize, what we found is that models that come from metastatic tumors from real patients, making those models is really challenging, but really might be what it takes to better understand metastatic breast cancer in humans. These patient-derived models do nicely represent the patient's tumors, and they can be used to predict drug responses. But we still have a lot more work to do to see if this approach of functional precision medicine will make a difference to patient care. This will require not only the feasibility trial that we're doing now, but also randomized efficacy trials on a much larger scale to see if this really works to improve outcomes of patients. But we're doing the first ones now, so we're very excited about that. With that, I would like to acknowledge the people who contributed to this work. So in addition to Brian Welm and our joint labs that really work pretty much hand in hand these days, all the people who contributed to this particular work are shown here in bold. I also want to thank especially the patients who participate in our studies and our clinical collaborators who, without them, we really couldn't try to make progress in clinical treatment of breast cancer. And of course, I want to thank all the funding agencies, especially the Huntsman Cancer Foundation, who funded this crazy idea way before we could get funding from federal agencies for it. And it has led to us being funded federally for these types of studies. But as I said, we really have a long way to go. So with that, I would stop, and I'd be happy to have some questions or a discussion if there are any.
Thank you, Dr. Welm. I hope you can all agree that what you just heard is something really special and extraordinary that is happening at Huntsman Cancer Institute. And only thanks to the doctors Welm at a few other institutes now in the United States. We knew, as I said at the beginning, that we brought in the superstar team when we were able to attract them to our institute, and they really are making an important contribution for the world in understanding metastatic breast cancer. We do have some questions. I'm going to ask you those questions, Dr. Welm, that have come in through the chat, and then we'll have you discuss the responses. The first one is, can biomarkers be used on initial diagnosis to determine if certain breast cancer therapies will be effective instead of waiting until it comes back as metastatic?
Yeah, that's a great question. So yes, there are standard biomarkers that are used on diagnosis, and those really are the three I mentioned. So estrogen receptor, progesterone receptor, and HER2. And that dictates what the therapy will be. So if the hormone receptors are there, that means the person will benefit from hormone therapy. If it's HER2, they will get Herceptin, and that will happen either before surgery or after. What we don't know we don't have any other biomarkers beyond those. So one of the things we're working on is, you can imagine it's not going to be feasible to grow every single person's breast cancer in a mouse to say whether or not that tumor might come back and might need more aggressive therapy. So we're working on a biomarker there that would say, okay, what's the molecular signature of a tumor that can grow in a mouse? So we don't actually have to grow it in the mouse to get that answer. So that's something that we're doing now in kind of a large collaboration, actually.
Thank you. The next question is, what determines the number of chemotherapy appointments that a breast cancer patient might need?
Well, the number of chemotherapy, we call this lines of therapy, is really variable. So the standard, if it's a standard breast cancer, the ones that are curable and don't come back, and not even everybody gets chemotherapy, because some people can be cured with just surgery and some people can be cured with just hormone therapy. But if you get the chemotherapy, it's typically just 1 or 2 lines of therapy either before surgery or after. And then hopefully there's no evidence of disease and you never need more. But if the tumor does come back, if it's ER positive, there's more hormone therapy combinations that happen. And it's not until after all of those are exhausted that you would need chemotherapy. If it's a triple negative breast cancer or a HER2 positive, those tend to need chemotherapy earlier after metastatic recurrence, but it's completely variable how many lines one needs. Basically, until you keep giving it until you can find something that works, and then you give it as long as you can. And sometimes there's tolerability issues because these are toxic drugs. So sometimes people have to come off simply because they can't tolerate them anymore.
Thank you. And I think that speaks to the overall emphasis of your discussion, that every cancer is different, even when we lump it into the same category. The next question is, in your opinion, what are the most common causes of breast cancer?
Oh, that's a good question. We don't really know the causes of most breast cancer. So there's a small fraction of breast cancer that we know is genetic. So that would be people who inherit mutations in BRCA1 or BRCA2. Everybody's heard of those. There are other genetic variants also who contribute to breast cancer. But for the most part, the rest of breast cancer, the vast majority, we call sporadic breast cancer, and we don't know why they happen.
As a follow-up to that, they wanted to know, do you have lifestyle recommendations to help prevent breast cancer?
I get this question a lot, but I'm not an epidemiologist. And so I don't really have the right expertise, but I do know that in general, breast cancer diagnoses are associated with the same kinds of things that lots of cancer diagnoses are associated with. So, obesity is a big one. Smoking actually is a big one. So all those, any healthy lifestyle seems to protect from all kinds of breast cancers. All kinds of cancers, including breast cancer, but there are many people with very healthy lifestyles that still get breast cancer. So it's not really something that's totally controllable.
Thank you. One questioner asked, do we treat men and women differently when they have breast cancer?
That's a good question. Again, I'm not a physician who's doing treatment of patients, and so I don't have firsthand knowledge of that, but I do know from the literature and from sort of anecdotal cases at HCI that the treatments are pretty much the same. So men do develop estrogen receptor positive breast cancer, and they do get things like tamoxifen or the same hormone therapies that women get. One thing that many people don't know is about estrogen biology is that actually estrogen in the body is made from testosterone. And so and that actually comes from fat. So, so the way that the hormones are metabolized, you actually, if you have a lot of testosterone, you actually can have a lot of estrogen as well, because it's a precursor to estrogen.
Thank you. There's a question about access to healthcare and disparities in providing healthcare for people who maybe for economic disadvantage where they live, race, might be factors. The question is, what is Huntsman doing to address those issues in accessing healthcare?
Yeah, that's a really great question and an important topic, because we do have minority and marginalized populations that they really do have worse outcomes. And there's two parts to that. A big part of it is access to care and things like insurance. But the other is actually the biology of the disease can be different in different populations, just like every breast tumor is different. But it is true that minority populations tend to have more aggressive biologies of breast cancer. So HCI has a big effort in community outreach and sort of equity and diversity in this area. So I would refer people to look at the Center for Hope at HCI, where they have a ton of outreach, not only into our Native American and Latino populations in the state, but also to the rural and frontier populations. That is actually a big area of disparity in the Intermountain West, where people, may not be of an ethnic minority, but they may have still just as reduced access to healthcare and health education and things like that. So that's another big area of need.
And tell me a little about the mobile mammogram service that Huntsman offers.
Yeah, so we have the mammogram bus, which is this really huge bus that actually is like a mobile clinic, and it's just amazing inside, as far as like it's the fanciest clinic on wheels that I've ever seen. And I don't know its actual schedule or where it goes, because it's all over the place all the time, but it does go out into the community to provide mammograms to people who might not be able to travel to the institute or to any of our affiliate institutes to get screened. But it does help increase screening and access for many populations.
Thank you. There's a question about a perception that perhaps Asia has better breast cancer rates and cancer rates in general than Western cultures. First of all, is that correct? And second, do you attribute that to diet?
It's a good question. It's not one I don't have any authority to answer that, because again, I'm not an epidemiologist, but I do know that the incidence is lower in Asian and Latino populations than in white and Black populations. So we don't know the reasons for that. Again, we still don't really know the causes of breast cancer. So trying to attribute it to environment or diet or any of those things is tricky. It's like in all biology, I'm sure it's a combination of genetics, lifestyle, environment, et cetera. But it is true that the incidence is a little lower. I showed on that graph that has the progress over time, the reduced mortality over time. And if you remember, the white and Black populations were at the top and had a pretty decent downward slope, and the Asian and Latino populations were lower, meaning there's lower incidence, but their slope of so-called improvement, so less mortality over time, was a little flatter. So that suggests that we still need to make more progress in treating those diseases, the metastatic forms of those diseases.
Okay, this is my favorite question because it's so self-serving, but somebody actually did ask this. I'm not making this up. How do we support your work?
We receive a lot of wonderful support from the Huntsman Cancer Foundation. That's probably the one we count on the most. in terms of having a reliable, source of funding for these sort of crazy ideas that we have that really can transform, we hope, the field. So maybe Susan, you can tell them how one donates to the Huntsman Cancer Foundation, but we've also had support through, for example, Five for the Fight, which works with the foundation, and Qualtrics and the Utah Jazz to raise funding. So even just doing your $5 at a jazz game really does help a lot. We received, actually, we launched our second precision medicine trial with some of the Five for the Fight money that came from the foundation.
Thank you. And to give to Huntsman and support Dr. Welm's work, or for that matter, any sort of work that you might have an interest in cancer, you can go to huntsmancancer.org and click on the donate button, and you can put in the notes restricted to the Welm Lab, and that way 100% of your donation will reach Dr. Welm and support her work. Likewise, if your disease interest is in a different area of cancer, you can customize your gift and indicate that you'd like the gift to go to, for example, bladder cancer or skin cancer. And 100% of every donation made by the public actually goes straight to support the programs at Huntsman, with no amounts for administrative costs for the operations of the Huntsman Cancer Foundation, thanks to generous support from John Huntsman's family.
Mm-hmm. It's amazing.
I don't see any other questions. So Gabby, sorry, the note went away. Our real facilitator, not me, but the person behind the curtain, was sending me an instruction. And Dr. Welm, just to recap, I want to thank you again. And I want to I was taking some notes while you were talking, and here's the story from 2007, when you came to the present day. And it's extraordinary. You brought a technology that allows us to do a brand new xenograph, that allows us to study new ways to see how breast cancer is metastasizing, and that was through mice work. You said, well, then we can use this information for greater impact in cancer by, in the old days, we had a trial and error, somewhat selection of therapy, and today we can be more precise and identify, using the xenograph information, about what's the best medicine to give that patient. Medicines are hard to tolerate, you noted. A lot of them are toxic, and also you don't have much time. This is urgent. And so you talked about how you've sped things up. You used that information to then a new term. I don't know if you created this term, but I love it. Functional precision therapy, to look at things that are really going to work in one individual. And like we've said, every person's cancer is different. And what I love about that is a virtuous cycle of good that every patient is giving back through the generous gifts that they give, maybe financially, but think about it, they're giving of themselves to help inform the future, better treatments and outcomes for cancer patients. You noted that all this work made Huntsman Cancer Institute really stand apart in the field of breast cancer in the United States and in the world. And I remember when other centers started contacting us and asking us how they could get access to Dr. Welm and her tissue and the knowledge. And so you really have helped improve the reputation and improve outcomes. And then I love, the final thing you said of the work you're going back to better predict how aggressive a tumor is going to be and pick off those, you call them bad actors, at an earlier time, so that if additional time is what we're hoping to accomplish in the short term, we give people the gift of time. In the long term, of course, the holy grail for all of us who do this work every day is a full cure. And so thank you for being such an amazing part of the future of cancer treatment and hope for men and women who are affected by this directly, and for those of us who love them and lost them and want to see it change for others in the future. Thank you again.
Thank you very much.